inviting me to come over here in this beautiful coma to do a talk. Still morning. Good morning, everyone. I'm going to talk about the diagnosis of scabies in the laboratory, which is by PCR. As you've heard already in most of the talks, diagnosis of scabies is really challenging. Because in the early stage of the disease, in the early stage of the disease, it mimics all other skin conditions such as eczema, psoriasis, dermatitis, and a presumptive clinical diagnosis is based, of course, as you have heard earlier, on nocturnal pruritus and location of the papules, and of course your history of exposure with infected individuals. Microscopic examination of skin scrapings often fails, as mites are very elusive and they're very low in numbers. In the early stages of the disease, they're like you can find from 1 to 15. So they will really escape. And why are we concerned? Misdiagnosis always lead to improper treatment, serious disease sequelae, and increased chances of epidemics. And of course, increase in misdiagnosis results in inaccurate assessment of the effects of intervention and leads to failure of your control programs. So what are the tests that we can do in the lab? We always go for skin scrapes. We use our scalpels, scrape off the suspected lesion, and we use the microscope. We look for this elusive mite, or you can use Scotch tape, dab it with the lesion, and then just put it on a slide and have a look. Those are quick and easy and very sensitive because you have to sight the mite. You can also do some ink, put them in the skin, rub it up with alcohol, and again, look for the barrels. You can check for antibodies, but this is quite insensitive and has lots of cross-reaction. And of course, you can do PCR. Other tests that are available, clinicians, dermatologists, they would use a dermatoscope. But the resolution is like low, it's 10x. Higher resolution is by video dermatoscopy. And in high-end facilities, you can do RCM or OCT. For PCR, there's actually a few variations of the PCR. The traditional or conventional one is you extract DNA from your sample. You can do amplify or make several copies of it, and it's quite tedious because you have to look for these results bands in agarose gels. The one that offers a more streamlined workflow will be your qPCR, and it gives you some quantitative readout in terms of CT values.
Can you just explain what's PCR? Because I think a lot of people don't understand what.
Yes.
Thank you.
PCR is, remember when we had COVID, we get all the swabs and they put it on a PCR instrument. What it does is, from a DNA of the mite, it makes several copies so that you don't see the mite, but you can have mite material by the sample that you collect, either by skin scraping, swabs, and then the scabies genome or human genome, the genetic makeup of the mite, you amplify a portion of that, and to amplify that you need an instrument like a PCR machine, and you design primers to start the amplification or making several copies of it, and then you need a probe too so that you increase your sensitivity. And you use a little machine and you stick your PCR mixes in there, and what you see is a visual representation of your results. Like the standard PCR, you can see this in a gel, these bands. So it means this is a positive for scabies mite DNA and this is negative. So there is a visual representation of your result. And that is polymerase chain reaction. It's just basically making many copies of your mite DNA for you to be able to see if it's positive or negative. Is that correct? Okay, another variation of this, that's conventional, I call it, and it's endpoint. You know, after making several copies, you see the end. But there is another variation of this PCR that you do the same thing. You extract DNA, and then you put it in an instrument using your primers and probe to start the reaction. But it doesn't give you this gel, but instead it gives you quantitative results. And you see graphs like this, and then you know you have to use a standard curve so that this is your positive control reference. And you have this CQ threshold is the starting quantity. A threshold that says above here is a positive result and below here is a negative result. So it gives you numbers actually. And these values is synonymous to the number of copies that you have put in your little tube from the DNA that you have extracted, the genetic material from your sample that you have collected. So it gives you an amplification readout of how it happens in real time. That's why it's called quantitative polymerase chain reaction. It's real time variation of the PCR. For our group, we have developed a qPCR assay that you don't need to use the scalpel blade, so you make your sampling methodology less invasive. We use instead a swab, similar to your COVID swab, except that this is bristle. You know, if you can look closely with the bristles there. This is to increase the chances that you can scrape more mite material if they have bristles. So it's called flock swab. And we have optimized also a methodology that you can just buy a commercially available kit to extract your mite DNA. But then again, we went to the literature and we found a type of gene, which is mitochondrial gene. It's called in the Scabies genome. And because normal PCR, we have 1 target, 2 targets, 3 targets. You know, this is the subject of your amplification or making several copies of the target. What we did was to look at the whole genome of Scabies and then found a more abundant target because we want to make our assay more sensitive. And this is how we came up with these 2 other targets in our assay. The SSR5 there at the bottom and then the SSR6. So altogether we used 3 targets to make our assay more sensitive. And we've also gone through all the tasks of looking if our PCR will be highly specific. So we tried to get DNA from other mite species like your house dust mites that bring sneeze. So we got DNA of this, amplified that, some sheep scab mites, or there's also eyelash mite in humans. So we got this DNA from this. And then we also got head lice DNA, related species, and some common skin pathogens as you find with scabies, your staph, your strep, and then some other fungal pathogens that can be found in the skin. And then of course, normal human skin. So we amplified this, and of course it's highly specific. Our assay will only amplify DNA from scabies mites. They're all negative, so 100% specificity. And we also collected some samples in Darwin from the Darwin dermatology of some patients presenting with dermatitis, eczema, psoriasis, and some other fungal infections. And these are all negative as well among these patients with other skin conditions. So it's 100% specific, targeting only scabies mite DNA. For sensitivity, at the moment, our limit of detection is 3 copies of each target in the reaction based on this standard curve. So after optimizing our PCR, we went to look at the patients admitted to Royal Darwin Hospital. So these are clinical samples and they're admitted. We tried to test our PCR with them, and then when we were happy with the sensitivity with different clinical profiles, we found out that the 3 copies per microliter may not be capturing some of the other very low infection. So we went to, again, develop or design a new standard curve. Apart from the positive control being diluted tenfold, we now diluted them twofold, so the control is now giving us, based on the CQ values that we can now detect, our limit of detection of our PCR now is less than 1 copy of the target mite DNA per microliter. So it's really, really very sensitive. So we tested this in the Kimberley, and from the survey of kids in endemic communities, we found 20% positivity rate, and we found that really high CQ value means low, very low copy of DNA. So it's really less than 1 copy in field samples, like the 1 samples you collect by swabs in the community. And we've also tested this qPCR with the Auckland kids, 23% positive rate, and you know that it's variable in Auckland because the lowest copy is 0.9 copy of target DNA that we have detected here, but it certainly helped Simon and the university group to really suggest that there is currently Scabies misdiagnosis in educational institutions in Auckland. More recently, I came across this paper that reviewed all the 4 methodologies of skin scraping, adhesive tape, dermoscopy, and PCR. And these are published, trials from 2000 and the present. This is the latest meta-analysis. And they found out that average sensitivity is higher as you go through all the methods down to PCR at a level of sensitivity of 81%, and specificity is comparable. Having said that, we're thinking of how else we can improve our PCR diagnosis, because in the real scenario, you will have really very low level of mite material that we can put in the PCR machine. So the very latest technology in PCR is, there's a company that put up digital PCR. It's still a variation of the qPCR, but it certainly makes my life easier that everything is automated. And I don't need that standard curve, because the problem with the standard curve, your mite material should fall within the curve, or otherwise you might have a false negative result. It should fall within the control dilutions. And even if I say we can detect less than 1 copy per microliter, we still wanted another technique that we can increase our sensitivity more at very low targets. And I can, the 3 targets that I'm talking about, the SSR5, 6, and COX, you can put them in 1 tube and amplify them all at the same time. And that is called multiplex format. So if you have 1 sample, you don't only query or ask amplification by PCR with 1 target, but you can make PCR or amplify those targets simultaneously. So I tried my hand on this, on some samples from Auckland that Simon and group collected. And these are the preliminary or initial results. The instrument software gives me a visual representation of my results. As you can see here, I tried to amplify SSR5 on the top. The second one is SSR6 and COX. You can see that this red line gives me the dividing line between positive and negative. So these are my controls, and you can see the positive partitions in a 1D scatter plot. I can clearly see And these are the negative partitions, which means the relative results. Also, these are originating from the samples I have amplified for the 3 targets. The software also gives me exact copies of the target that I PCR'd up in terms of concentration, and it gives me copies per microliter. As you can see, the 3 controls have high copies, but when you go to the sample here, it's less than 1 copy per microliter. 0.4 here and 0.4 here in this sample, and that's positive. And if I go to this column, if you use a nanoplate where you do the reaction and put it in the machine, you can choose between 8,000 partitions or 26K partitions. When you do the test, this is equivalent to your number of replicates or how many replicates you try to use. So I've used an 8,000 replicate plate here, and it gives me the valid partitions in this column. The distribution of positive results or partitions and negative. As you go down here, only 1 positive partition is positive here, giving me less than 1 copy of 0.442 of Scabies mite DNA in the PCR. And here, it gives me also 0.429, and 1 is on the target SSR5, and the other one is a target of COX. So this is really, we have done the very, very highest level of sensitivity at this point, and I'm really happy with this. So I just need to run and confirm all the other samples that I have from Simon and Group, from the Auckland kids. So to summarize, yes, we have high technology qPCR for the diagnosis of scabies, but I will tell you it's not a magic bullet. It is a useful adjunct to help improve the diagnosis of scabies following the IACS criteria. And certainly this tool can be limited by your sampling technique. So we really rely on clinicians, field workers, in choosing the right site of sample collection. And also, we have to come to an agreement to standardize how we design our sampling protocol. And we always have to look at the history of treatment. If they've undergone treatment in the last 2 weeks, then we won't find any more mite material there, and we'll give you false positive results. And also the history of contact with family and any individual. But definitely, PCR gives you an objective and definitive diagnosis. We have an ongoing collaboration in University of Auckland's case control study, trying to look at the relationship between rheumatic fever and scabies, and we're putting in all these technologies to help improve the diagnosis. And we'd like to thank the funders, HRC, and in the early stages of this work, we're funded by the National Health Medical Research Council of Australia, and these are all the collaborating institutes that would collect field samples for you to be able to optimize a highly sensitive and specific qPCR diagnosis for scabies. Thank you.